next topic, there's a slight change because Dr. Rajesh Pande also has to catch a flight, and he will actually be delivering the next talk. And Dr. Rajesh Pande will be talking on a burning topic today on antifungal resistance, which is an emerging threat. Dr. Pande is the principal director in HOD at BLX Max Super Specialty Hospital, and he's been the former general secretary at the ISCCM, and to his credit, he's been the organizing secretary at the Criticare 2023, and he has to his credit publications of over 56 contributions in the book chapters over 20. So may I request Dr. Pandey to deliver his talk. Thanks.
Thank you very much, sir, for the kind introduction. So I'm here again. I'll talk about antifungal resistance. We are more concerned about antibiotic resistance, but antifungal resistance is also increasing, and more or less the factors are same. If you see the classification of mycosis, we have superficial infection, subcutaneous infections, and systemic infections. So all the infections that we encounter in our clinical practice in the ICU domain are systemic, Candida, Aspergillus, and mucormycosis. And generally they will, enter the body through a deep focus in internal organs like paranasal sinuses, digestive tracts, or lungs. And the infection may often start from the lungs and then it will go to other organs. We have a huge global fungal burden. 300 million people are affected each year globally, and there is a high mortality. In US, they are spending about $7.2 billion in the care attributed to fungal infections. That was 6-7-year-old data. And most common fungal infection in our domain is invasive candidiasis in India, which is followed by invasive aspergillosis and invasive mucormycosis. Cryptococcosis, which has been identified, Cryptococcosis neoformans, as a major threat, critical threat, is a rare infection in India. So CDC for candidemia has estimated a crude mortality of more than 25%, which is 40 to 90% in aspergillosis, especially if they belong to the immunocompromised group. And 30-day all-cause mortality reported for this is 43.5. And so the mortality burden is high. If you see this famous epidemiological study by Arunlok Chakravarti et al., we are developing fungal infection in patients who are less sick, where Apache score is less. And we are also getting significant high number, 80% is non-albicans. So that is another problem. And we also are getting more and more reports of Candida auris and Candida rugosa. So that is a problem because this is generally resistant to many drugs, and azole resistance is increasing, and there is multi-drug resistance also being reported. So it's a problem. This is an old CDC poster which is highlighting about drug resistant species, and they found at that time in 2019 or 17 that about 7% of the blood infections were resistant to antifungal drugs, Candida related. So it's a problem there. Second bug is invasive aspergillosis. Now we know that this is more commonly reported from in the hematology domain, HSCT and hematological malignancies. But in India, it's being reported more and more in cirrhotics and COPDs, not so commonly reported in hematological patients as is reported in the Western literature. So again, the mortality is significantly higher if the patient is neutropenic. So we have to keep in mind. Third infection would be invasive mucormycosis, invasive mold infection. Now we saw this in what happened in COVID. It's a fungal infection, no biomarker is available, and oftentimes the lab is confused. They can't distinguish from aspergillosis, and nosocomial outbreaks are commonly reported in India. The mortality without treatment is 100%. With treatment, possibly it will come down to by half. And risk factors in North India are uncontrolled diabetes, post-tuberculosis, and in South India, CKD. Now, these are the various risk factors that have been reported for invasive candidiasis, aspergillosis, and mucormycosis. So this makes the patient susceptible to develop these fungal infections when they come to our ICU. If they stay for a long period of time, if we are using more invasive lines, doing more interventions, they are likely to develop these infections. Now, concerns in India, specific concerns, Candida auris is being reported as a predominant non-Candida albicans species. Two hospitals that have reported that this is the number one Candida non-albicans, Dinanath Mangeshkar and Gangaram Hospital, Delhi. So that's a problem because it is highly resistant to azoles and pollens, and the mortality rate is high. Like I said, invasive aspergillosis is more commonly reported in COPD, liver failure, and cirrhotic patients. And mucormycosis is 70 times more common than what is in the Western world. So that we need to remember. So this is what WHO says, the fungal priority list. And you can see our bugs, Aspergillus fumigatus is here, Candida auris is here, Candida albicans, I would consider it less of a problem in India because we are now a non-albicans country. And then the high priority, you have two of the non-albicans, Tropicalis and Paracellosis. And this is the medium priority list. Now, if you see what are the drugs that we have, most of the drugs that we use in our clinical practice are either azoles, echinocandins, or pollens, and some other drugs. So the armamentarium is limited. The development started post 2000 when the antibiotic pipeline became dry, and uh, the drugs are developing resistance now. So let's see this. Now this is showing the drugs that are used. So we have, this is the fungal cell wall, this is the fungal cell membrane, and let's talk about echinocandins. Echinocandins will work on the cell wall and they will inhibit the beta-1,3-glucan synthase and disrupting the cell wall integrity, leading to severe cell wall stress, and generally mediated by, in Candida, Cryptococcus and Aspergillosis by FKS1 gene, and in Glabrata, etc. By FKS2 gene. So here what happens is there is modification in these genes, so these drugs cannot work. So that we need to understand. And then the second would be the azole. Azole, we know that it works on the cell membrane, and this will inhibit the synthesis of ergostrol by inhibition of lenostrol 14 alpha demethylase. Here you can see this. And this results in, because ergostrol is not being produced, so there is a lot of accumulation of toxic sterol. You can see it here. And this again, this generally by using ergostrol 11 gene. So the modification will occur in ergostrol 11 gene, ergostrol 3 gene, and that is how the drug becomes ineffective. The third important antifungal we have is pollens. Pollens we know are like sponge. They will suck out the sterol and will create holes, and there is ionic leakage leading to cell disruption. And the resistant mechanism is by ERG genes. So the antifungal cannot work. It's similar story like we have seen in the antibacterials. What happens there? And here you can see, if I have to summarize the mechanisms, there is biofilm formation like you have in bacterial infections. There is loss of mitochondrial function. There is stress signaling. There is mutation of in the cell wall biosynthesis genes like I explained. And there is inhibition of pyrimidine salvage pathway, increased drug efflux. Same like you have the antibiotics thrown out, and upregulation of mutation of target genes in ergostrol biosynthesis and lipid metabolism bypass. So these are the predominant mechanisms that you see. And if you look at what is happening overall, now this is a very nice paper published in Science. So you can see that for azole, the resistance status is, we are possibly a lot of resistance. It is completely gray. There's no antifungal where we have no resistance. And the resistance is present in humans, in the animals, and in the plants. Please understand, antifungal spray, antifungal drugs are being extensively used in agriculture. So that is where the problem is. And so many of the mechanisms we may not even be knowing. So there is an interplay of all these. This is one fungal species reported azole resistance by country. So here you can see this is in the plants. From 1997, you see in 2017, the resistance has increased. Now, this is talking about all these bugs, the Aspergillus fumigatus, Candida albicans, aureus, glabrata, etc. So in humans also, you can see that it is increasing in intensity over a period of 10 years or 20 years. So this is a problem which is increasing. And if you look at why it is happening, the fungi in the environment are being exposed to broad spectrum antifungal agents because, as you can see, the climate is changing. There is global warming. We have more and more therapeutic interventions available, sicker patients, more treatment options for cancer patients. So you have susceptible group, prolonged ICU stay patients, and you have antifungal being used in the agriculture largely. So the virulence of the fungi is also changing. And all this is creating environmental hotspots. And because even the soil is getting infected with these resistant mechanisms, antifungal mechanisms. And then you have the human exposure. And finally, there is a complex interplay. Same problem as we tell in antibiotic resistance. So the drivers of fungal resistance are crop destroying fungi will account for 20% crop losses worldwide. So systemic antifungals, fungicides are frontline treatment for fungal disease in humans as well as in plants. And 9 times more antifungal compounds are available to control crop disease than to treat the systemic animal infection, which includes humans as well. So fungal pathogen can rapidly develop resistance because of this exposure. And in humans, if we are giving long period of prophylactic treatment with antifungal agents, then it can lead to, there's on one side, good side, it will prevent the emergence of infection in this high risk population. The other side, there is a risk of emergence of antifungal resistance. So if you look at the evolutionary drivers, again, the paper from Science, the population is increasing, urbanization is increasing, and there is monoculture cropping. That means you want to grow one type of crop only, which is susceptible to fungal infection. Then you use antifungal agents there. So you have susceptible host plants, and then more immunocompromised patients, more aggressive cancer therapies are available. So you have a vicious cycle where there is global dispersion of these resistant strains because the world has become a global village. And finally, you have, patients who are having long courses of prophylactic drugs or repeated treatment of the plants with the same fungicidal. So this is leading to a vicious cycle where the fungal resistance is rising. So what are the strategies that one can use? Well, it has been suggested that uh, when you encounter resistance, instead of monotherapy, you can go for combination therapy. The problem is for antibacterials, if you have carbapenem resistance, you can use your either this CARBA test or BioFire, which can tell you some of the ESBL mechanism of action. I haven't seen any, at least my lab in Max reporting about the antifungal resistant mechanism. So if you have to combine any two antifungal agents as suggested here, maybe you have to use your clinical acumen and see, and maybe check the antifungal antibiogram and antifungal, the response of last year's data. So it requires a multi-pronged strategy. It requires environmental fungal AMR strategy, patient fungal AMR strategy, and nosocomial fungal AMR strategies. So the pathogens in the environment are Aspergillus, Candida, Mucor, etc., many of them. And then in susceptible individuals, again, Candida glabrata, Aspergillus fumigatus. So the idea is we need to understand that there is a complex interplay. They are available, they are there in the environment also, in the soil. And then intervention could be that environmental antifungal stewardship should be started. And antifungal stewardship in humans, and then national antimicrobial surveillance program. Unfortunately, we do not have that even for antimicrobial resistance, the non-fungal group, bacterial group. So it's been said that, what can be done as an ideal model, fungal samples can be acquired from the clinic and environment, and using the traditional cultures, they can be grown, and then subsequently the genomic study can be done, which can be pooled, and then the scientists can analyze, they can identify the hotspots, do the surveillance, and, help. So antifungal is resistance is an emerging problem. We are becoming more and more invasive in our approach. More and more therapeutic modalities are available. And, earlier we used to say that probable, possible, proven kind of fungal infection. Now we are saying that prophylactic, therapeutic, and what do you call that? The two other things where you base it on the biomarker. So the classification is changing. You want to use it early, the antifungal agents. So the thing is changing. We need to be aware that antifungal resistance is increasing, and we need to be prudent when we are using them, rather than empirically adding antifungal just because we feel that we need to add something. Thank you very much.
Thank you, Dr. Rajesh Pande. That was a very lucid presentation. I think Sir has to catch a flight. So if there are any questions from the audience, I think he'll be happy to take a few. He's very well highlighted the factors causing the antifungal resistance, right from agriculture, climatic changes, and the use of dual antifungal possibilities. So, any other questions? Please feel free to ask him.
Dr. Pandey, especially the immunocompromised patients, any role for altering the gut microbiota?
Any role for?
Gut microbiota?
Gut microbiota actually has only been explored for C. diff infection so far, fecal transplantation. For fungal, I do not know, but I think what we know is that the human microbiome is changing in disease conditions. So the American Gut Microbiome Project showed it, that in different disease patterns, even in sepsis, you have a different pattern. How that will be effectively explored to provide us some solutions, I do not know. So far, only fecal transplantation in C. diff is there.
There are no further questions. We'll go to the next topic.